I want to say a couple of things just slightly off message here. The first one is that the other thing that you learn with these nominations is something about yourself. You know, you start thinking about why you do things and why somebody else has done it differently and what you might learn about yourself and your own science in the process. I really enjoyed it, and I utterly recommend sitting on one of these committees. Well, I won't say they're the most fun you can have with your clothes on, but I just did. We're going to save the best for last here, both figuratively and literally. The Yentsch-Schindler, you know, there's a bet on apparently whether I drop my notes. The Yentsch-Schindler Award is named after two of the most iconic scientists in our discipline, and it's intended to honor early career scientists, those within 12 years of their PhD, who have demonstrated a dynamic but yet balanced approach to research, training, that is mentorship, and service to the society. And today's awardee, Matt Church, is described in his letter, but if you've met him, you'd know he's the embodiment of this. Before the awards, I asked Matt, well, you know, what are you most proud of? And often we roll out, well, it was my nature paper or whatever. Matt said, my teaching. And I thought that told me a lot about him. It told me about, you know, how he values his role as an educator and how he sees he's going to move science forward in the future. His own education, he has a master's and a PhD from College of William and Mary, where he worked with Hugh Ducklow. But most recently, over the last 10 or so years, he's been working at the University of Hawaii in a number of different capacities. First as a researcher on the Hawaii Ocean Time Series program, then as a director, and then assistant, now associate professor with tenure. Matt's, I guess if we're starting with service, this is where he's really come to the fore in promoting this program, but also in developing the Seymour program, which is an international program to train microbial ecologists in both research and education. And this is an incredible achievement. At the same time, in less than 10 years since PhD, he's managed to publish over 60 papers in terrific journals right across the board on diverse topics. I can't even list all the topics, but I mean, the one that immediately jumped out to me is, oh, new diazotrophs. God, is this something I've got to figure out for lakes? You know, it was just really inspiring amount of science and education and dedication. So I'll keep it short here, but I just want to say that one of the things that was evident from his nomination, and which when I looked through his CV earlier, that jumped out at me, was we often hyper-specialize. We're forced to do this because of funding, we're forced to do it for visibility or whatever reasons. And Matt shows that you don't have to do this. You can be broad-based science, you can dedicate yourself to education and also to mentorship. So with that in mind, I'd like to welcome Matt Church to the stage to accept the 2015 Yentsch-Schindler Award for Early Career Researcher.
Thank you very much, Peter. This is, as you might imagine, a true honor for me, and even more so because David Schindler is here in the audience, which makes it a bit intimidating, but also a real pleasure. So I want to thank all of you for coming today. I know we're at the end of a fantastic meeting, and I want to acknowledge the meeting organizers for putting us all here in this fantastic place, and so thank you for that. Of course, in my 15 minutes here, I want to acknowledge a lot of people. I want to use this as an opportunity to acknowledge people that have really helped promote my career and created opportunities for me. I'll tell you a little bit about some of the science I've done, and hopefully, maybe if there's time, tell you about some lessons I've learned along the path of my fortunate path through microbial oceanography. I have this quote up here. When I was young, my dad, as a scientist, used to tell my brother and I to live the questions, which at the time I kind of assumed it was because he didn't want to answer our persistent questions. He would just say, just live the questions now. And I didn't understand what he was talking about. It wasn't really until I got into studying science in college that I realized that the process of doing science ultimately leads to more questions, and it's really that process that I think is the important thing for us to remember. So I want to say thank you to the award selection committee, because after coming to this meeting and seeing the fantastic talent pool amongst young scientists in this society, it's truly an honor for me to be here. I want to thank the ASLO board for having the foresight to create this award in the first place. When I met Jim Elzer on Sunday, he said the award was intended to be empowering, and it certainly is doing that. It's often easy as a young scientist to get a bit overwhelmed by all the responsibilities that are thrust upon us, teaching, mentoring, research, writing papers and proposals, et cetera. So having this recognition is really important. And then I want to thank Helen Schneider-LeMay and her team for all the logistics in making this meeting happen. I'm going to tell you a little bit about the Hawaii Ocean Time Series program and some of the science we're doing in the HOT program, but before I do that, I just want to acknowledge some of the people that have been really instrumental in keeping this program alive. And I guess one of the resounding themes of what I'm going to tell you today is that I think time series programs, in particular ocean time series programs, are really critical to our understanding of planetary change. And those time series programs wouldn't exist if it weren't for the dedicated effort of a lot of people. The HOT program has been running since October of 1988, going on 27 years. And many of these people that are shown up here have been involved in this program since nearly the time it started, spending a week at sea nearly every month of every year. And so it's an incredible amount of dedication, and so I want to thank them. There's also a number of people that I want to specifically acknowledge in helping my career. The first is on the far, I guess that's your left, my uncle Tom Church. Many of you may know Tom. He's a distinguished oceanographer in his own right, recently retired at the University of Delaware. And Tom had enough faith in me when I was bumbling around coming out of my undergraduate degree that he used his connections and got me into the Bermuda Biological Station for research, involved in the Bermuda Atlantic Time Series program. And that fundamentally changed my life. I had never done oceanography before, and so Tom really fundamentally changed the direction of where I am now. Hugh Ducklow, also shown here. Hugh is my graduate advisor and has been a tremendous supporter of my career, and I continue to really value interacting with Hugh. Dave Carl has another person that I consider a mentor to this day, and I've worked closely with Dave for almost 15 years now, and he's just created enormous opportunities for me in my career, and so I really want to acknowledge Dave. And then John Zare, also shown over here. John was my postdoc advisor and someone that I continue to collaborate with, and John has just been incredibly generous and gracious in sharing his ideas and helping promote my career, so I want to acknowledge John as well. And then, of course, my family. I wouldn't be here without their love and support. I was hoping they could be here to see me get this award, but they're at home doing the hard work, and I'm here enjoying this lovely place, so thanks to them. When I learned that I was going to receive this award, I thought for a minute about what I might say, and I didn't know David Schindler until breakfast yesterday, and even then it was really just a casual hello. I began reading papers by Clarice Yentsch and David Schindler to try and get a sense of the things that they stood for and still stand for to promote in science. And so I sort of tried to crystallize some ideas to share with all of you, and hopefully, I'm sure David will tell me if I'm wrong in some of these, but I wanted to make a few points that I think are relevant to the work that they've done. The first is to do science that matters, and not only matters to you as an individual, but matters to people outside of this room, the people that often pay the bills for what we do. And I think it's important to keep that perspective. And that leads to the second point, and that is engage people through your science. And I think that's a really important thing that I think about a lot, and I think it's an important message that whether it's through training of our students or our postdocs, or whether it's just engagement of the public, if we want to try and change the perception of the public through the science we do, it's really important that we engage them and get them excited. Take holistic views of ecosystems, and this is clearly something that both Clarice Yentsch and David Schindler promote, and I think is something that I work hard to try and better understand the complexity of marine ecosystems, to understand how the whole is greater than the sum of the parts. And then finally, teach and be taught, mentor and be a mentee. And this is something I work every day to try and be a better teacher, a better mentor, and I think, I hope that I'll spend the rest of my life finding people that I consider role models to be my mentor, and hopefully I'll be a student for the rest of my life as well. So when I got my PhD, I received my PhD in 2003, and I was really a product of the JGOFS program. We've already heard about the JGOFS program in the plenaries earlier this week, the Joint Global Ocean Flux Study. And I worked on measuring bacterial production and looked at controls on bacterial production. And really the idea was to try and relate my work to the role of the biological or biology in global carbon cycling. And at least I thought coming out of the JGOFS, so the JGOFS program ended at about the same time that I was getting my degree. And I think at the end of the JGOS program, we had a pretty good idea that we understood many of the fundamental processes that underlie the biological pump. That is the net movement of inorganic carbon across the air-sea interface through the action of biology, ultimately production of organic material, and eventually through a variety of processes, removal of that organic material from the upper ocean to the deep sea where it gets remineralized and maintains a strong vertical gradient in inorganic carbon concentrations. Since the end of the JGOS era and since the time I got my degree, there have been a number of really big surprises, I think, most of, in my own interests related to microbes. And those surprises really fundamentally change, I think, the way that we have to view the biological pump. And I would argue that we really haven't conceptually or quantitatively begun to integrate some of these discoveries into our understanding of the pump. And I've listed a few here. Some of my favorites include our knowledge that many of the organisms that we previously considered autotrophs, that is, in the upper ocean, those were probably phytoplankton assimilating inorganic carbon, using light as an energy source, reducing that inorganic carbon to organic matter. Those organisms probably, in many cases in the open ocean, are mixotrophic, in many cases assimilating dissolved organic matter, or in other cases perhaps even consuming other cells as a source of nutrition. And we really don't understand how that influences our view of the biological pump. Similarly, many of the organisms, the heterotrophic bacteria that we would have probably at the end of JGOS thought we understood their net role in this ecosystem, or in the open ocean anyway, we now know that many of these organisms have the capacity to use sunlight that presumably may have some influence on their physiology and metabolism. And in some cases, many of these organisms aren't heterotrophs at all. They're actually chemoautotrophs, and in fact, fixing CO2. And so our understanding of the complexity of the microbes in the ocean really led us to begin the course in 2006 as part of the Center for Microbial Oceanography Research and Education. We started teaching a summer course where we bring 16 students to the University of Hawaii for a 5 or 6 week period every summer, and we immerse them in concepts of microbial oceanography. So trying to integrate in an interdisciplinary way concepts about marine microbiology, biological oceanography, and biogeochemistry. And we like to think anyway that the students that have come out of this program are really going to be poised to be the leaders for the next generation in our field. So I want to tell you a little bit about some of the research I've been involved in, and the purpose of this slide is really just to convince you that time series programs are absolutely fundamental to helping us understand how we're changing this planet. And I think we all have heard throughout the week, and certainly we knew this coming into this meeting, that humans are altering the planet in many, many different ways. And I've just selected a few examples from the most recent IPCC report that includes on the far, on your left, is that right? Your left, the oceanic CO2 measurements as part of the Hawaii Ocean Time Series program. The red is showing the Keeling curve, the iconic measurements from Mauna Loa Observatory. And the point of this is simply that we know that the ocean is changing and changing in lots of different ways, but we wouldn't know that if we didn't have time series programs to give us time context to understand how things were before and how they are now, and help us to project how they might be into the future. So I got involved in time series programs initially through the Bermuda Atlantic Time Series study. As I mentioned, my uncle used some connections. He introduced me to Tony Michaels, and I went to Bermuda, and I had no oceanographic experience and had never even heard the word biogeochemistry. And Tony was such an energetic and transformative person on my life that he made me convinced that this is really what I wanted to do with my life. And so later I went out to Hawaii, where I've been for working with the Hawaii Ocean Time Series program for nearly 15 years now. And my work in both of these places has convinced me that these subtropical ocean gyres, not only are they some of the largest habitats on the planet, and they play a really important role in global carbon cycling, but the time series programs that we have there in both of these locations are really fundamental in our ability to constrain carbon cycling, both the magnitude of the fluxes in this system as well as pathways of those fluxes. So I wanted to give you a sense of what the upper ocean habitat looks like, and the resounding message here is that this is a persistently, now these are data from the North Pacific subtropical gyre, it's a persistently oligotrophic habitat. So, and that's maintained in large part by thermal stratification of the upper ocean throughout the year, and relatively high fluxes of light throughout the year. And so that results in very low inorganic nutrient concentrations in the upper ocean. And in contrast, rates of productivity, shown here in the blue, measured by the C14 technique, rates of productivity are always highest in the near-surface ocean where the light is greatest, which probably makes some sense, but we've spent a lot of time in the HOT program trying to learn more about the processes that supply nutrients to support that productivity. So I've shown here, I've just taken those same data and binned them by month to give you a sense of what the seasonal climatology looks like in this system. So in dark blue, the mixed layer depth, and you can see it's a little bit deeper in the winter when the temperature's cool, and then it stratifies rapidly into the spring and summer. Nitrate concentration, so the input of nutrients a little bit higher in the winter, completely devoid of nutrients basically through much of the spring and summer. And yet in contrast, rates of productivity increase seasonally, coincident with increases in light. And so that could just be driven by increased recycling in the upper ocean, but we know that's not the only story, and we know that in part because when we look at, so in shown in green, rates of productivity, next to that, particle export out of the upper 150 meters of the ocean based on sediment trap collections, and then in the bottom panels, in blue, particulate carbon export near the seabed, so this is now at 4,000 meters depth, and then in orange, biogenic silica fluxes to the seabed. And what we see is that despite fairly weak seasonality in particulate matter fluxes out of the upper ocean, at the seabed we see a very predictable and repeatable pulse of material that comes out of the upper ocean, and we know it's driven by the export of diatoms. And when we look at these diatoms microscopically and do genetic analyses, these are very specific genera of diatoms that recur every year, and those organisms have associated with them nitrogen-fixing cyanobacteria, which fundamentally sort of changes the way we view these ecosystems. In other words, perhaps subtle overprinting or shifts in the plankton community structure can appear to have very profound influences on the removal of carbon to the deep sea. So just as a few points of conclusion, I think we've learned from this program that food web structure and perturbations in that food web structure are really central controls on carbon cycling in this system. And I would argue that in order for us to mechanistically understand how those perturbations occur, we really need to better understand the ecology underlying that dynamic. In addition, we've learned that biological and physical processes interact to control carbon cycling, and we know that both of those both biological and physical processes are climate sensitive, and so that really demands that we study those processes in the context of time. So I'm sure I'm out of time, but I'm going to take one more slide to give you a few lessons that I've learned along my fortunate path. And the first is probably self-explanatory. I hope it wasn't directed to me, but from Debbie Bronk. And I think this is a good thing to keep in mind. In other words, I think it's good to be a good colleague and to strive to be a good colleague, and I'm working to do that. So thank you, Debbie, for that advice. I think it's really important that we work with fun, creative, and smart people. It just helps promote the interactions you'll have, and you'll do better science. I've quickly learned that time is your most valuable asset, and so use your time wisely, prioritize your time, and do things that are going to matter. But at the same time, remember to give back to the society, give back to the programs or people that have helped to advance you. And then finally, find your balance, whether that's in work or in life, and work to keep that balance. So with that, I want to thank the National Science Foundation for funding much of my research, the Simons Foundation for funding as well, my involvement in the Center for Microbial Oceanography Research and Education and the HOT program and the Ocean Carbon Biogeochemistry program. So thank you very much.